Good morning. Welcome to CU Anschutz, the University of Colorado Cancer Center, for this opportunity to commemorate Lobular Breast Cancer Awareness Day in Colorado. Thank you so much to our patient advocates for being here and to talk about what we're doing in lobular cancer with the Sikora Lab. Really cheers to the Lobular Breast Cancer Alliance for what they've done over the last year or so to organize the recognition of October 15th as Lobular Breast Cancer Awareness Day. Obviously, this is really a huge milestone in where we've come in advocacy, in where you all come in advocacy, in our ability to talk about lobular breast cancer as a separate type of breast cancer that deserves its own independent attention, and that you all need care that is different from other patients facing breast cancer. It's really exciting to see how many events worldwide are happening. This is the map that LBCA put together. There we go. Obviously, with Colorado being the most important star in this map, but it's really exciting to see how many sites worldwide are holding research and patient advocacy and clinical education events today to commemorate the event. So just briefly, what we'll talk about today. I'll give a brief introduction on my perspective on where we've come since I started research in this field 12 or 13 years ago. I will hand it off to Donna and Tracy to talk about Front Range ILC and their perspectives from the recent 2024 ILC Symposium in Europe. I will talk a little bit about some of the more exciting and translational research updates we've had over the last year in the Sikora Lab. And then I will hand it off to my labs. You can meet the people that actually do the work. I just write the grants and get to talk to you all. They actually do the work. And they'll talk to you all a little bit about what they're doing in the lab. I will talk a little bit about the Lobular Breast Cancer Research Fund, or the philanthropic fund that supports our research on campus, and what we can do with philanthropic support that's a little different than grant funding. Then we'll wrap up. A lot of Q&A throughout. Always feel free to stop me any point to ask questions. This is the fun part of this. And then you'll notice the lab's all got research posters up in the back, and they'll be around to meet and chat about their research and what they're doing. And then I'd be happy to take folks back to the lab to see where we actually do the work and have you actually take a look at some of the breast cancer cells and lobular cancer cells we have in culture in the lab that we study. So again, so my perspective on where we've come in community clinical and research advocacy for ILC. After I did my graduate work, I moved to the University of Pittsburgh with Steffi Oosterijk and Adrienne Lee, where they were starting a nascent lobular breast cancer research program. And in writing my first few grants in lobular, these are literally reviews I got on my first few grants around 2011 when I wrote my first few grants. The PI states that ILC remains chronically understudied, but this statement is not validated in any way. And my favorite one, the main weakness of this proposal is that ILC is a rare tumor. I think the vindication of this actually came, and again, cheers to LBCA for pushing this through, is NCI now actually has a separate page dedicated to lobular breast cancer and statistics on this type of breast cancer related to the SEER National Observation Program. And this is now my favorite part of this page right here. So now we no longer have to justify this. And for years now, it has not been the case that reviewers are pushing back like this, but really it speaks volumes to what the patient advocacy community has accomplished. This is not me doing this. This is patients and advocates being at the table at programmatic meetings and telling program officers, telling decision makers in grant agencies at NCI that lobular breast cancer matters. And obviously, we have a long way to go in research, but I think we did it from at least the first bar of the advocacy and awareness space. I think this is really exciting. I have QR codes up throughout, and if you point your phone at those, it'll take you to whatever page I have up instead of typing in everything over and over. So in the context, so I started my own lobular breast cancer research in Pittsburgh in 2011, and then I moved here to Colorado in 2016. So where have we come since then for lobular breast cancer research in Colorado since then? So the lab opened August 1st, 2016, but our first outreach event was held in June 2022 over at the Night Horse Pavilion. I think there were maybe 10 or so patient advocates that attended. Thanks, Ann. And that was supported by the BCCRF, which kind of got us our kind of kick in the butt we needed to start having these events. And that was an outstanding start to this group. We had a couple of events in 2023, including what I would consider the debut of Front Range ILC, where Tracy and Donna talked about what they hope to organize in the region. In fall last year, we held a science boot camp where we actually spent a morning basically going through the entire medical and graduate school curriculum on breast cancer in preparation for the Lobular Symposium that year. And obviously here we are today for Lobular Breast Cancer Awareness Day. So a few notable events in the interim, as far as us becoming a kind of an independent research lab and actually having dedicated funding to do lobular breast cancer research. I got our lab's first really multi-year dedicated grant for ILC in 2020 for the American Cancer Society. I think at this first outreach event, this is where I got to know Tracy, and Lori Petiti in California has been a longstanding supporter of mine. And this was, I think, really fabulous again to kind of start our outreach program. That was also a really good year for research. We got multi-year awards from the Department of Defense Breast Cancer Research Program and the NIH. So we're moving forward as far as getting independent funding to do lobular breast cancer research, that these grants are dedicated to ILC work. Between then and 2023, I met Donna and also Sue Corrales in California as my lab started to think about metastatic lobular breast cancer research. So they've been an outstanding help both with Front Range ILC and in thinking about my lab is kind of new to metastatic breast cancer research, how to think about it, what the needs of the community are, and how to write grants in that space. In the interim, we also lost Lee Pate, another one of my longtime advocate partners, but she helped establish the Lobular Breast Cancer Research Fund, and I'd be happy to help come show anybody who wants. Her plaque is downstairs across the bridge over there. So she is with us, and her name's part of our program forever moving forward at this point, which is amazing. I won't talk too much about this because Donna and Tracy will, but it is so exciting to see that the Front Range ILC Facebook group is now up to nearly 70 members. And it's great that this network is coming together across the Front Range. And in the interim, my lab is now up to 8 people, and you'll get to meet everybody here today. And it's really exciting that I have such an amazing, dedicated team. This is us at a casino heist escape room. We're not just like at a bank robbing it or anything. But it's been really amazing in the years since, both how our research has grown and how our work with you all has evolved in that same space, and I'm really excited about it. Finally, just a little bit on where research is nationwide. So this is a map of where, this is not like hard numbers, this is where I can find people publishing abstracts, getting grants, putting out papers on lobular breast cancer research, where there, what I would argue are probably fairly dedicated teams focused on the disease. It's not huge yet. Of the 71 NCI designated cancer centers, I would argue there are about 20 with really active ILC research groups, either clinical or basic. Again, don't hold me to it, I'm sure there's some missing. But this has grown over the last few years too, which is really exciting. I think what's also been a lot of fun for me is also seeing how we're growing to connect to other groups that are studying ILC nationwide. Like I said, I did my training in Pittsburgh with Steffi Österreich, so that remains a strong connection to their lobular breast cancer research group. Steffi is affectionately referred to by much of the advocate community as the Lob Mother, so I'm happy to be one of those descendants. I have a strong active collaboration with Becca Riggins at Georgetown and her lobular breast cancer research group. We have a number of papers together and are continuing to write grants. Our Department of Defense funding is actually collaboration with Julie Ostrander's group at the University of Minnesota, and she's helping us study lobular breast cancer tumorigenesis, the very earliest stages of lobular breast cancer. I also have an active collaboration with Jimmy Ray at Michigan, who I actually did my graduate work with, funded through the Dynamy Foundation to think more about estrogen receptor function in ILC. I have some research proposals that have been approved to get some clinical trial data with Dr. Rita Mukhtar at UCSF and her lobular breast cancer research program. And I've convinced Dr. Kevin Chung, a metastasis researcher at the Hutch in Seattle, to help write a few metastatic lobular breast cancer research grants with me. And we're just waiting for one to get hit. And actually a few pilot experiments have been cooking already. So really, I think it's quite like aesthetically pleasing that we are obviously the center of lobular breast cancer research work, geographically, if not literally, and it's really been amazing to bring together other groups nationwide to move research in this disease forward. Finally, just as a teaser, the two things I'll focus on today as far as where ILC research in Colorado is heading and kind of the most translational bits. This is actually a slide I showed at our first outreach event, and I'm really excited how this kind of offhanded comment has moved forward and what we were able to do in that space. And then if you'd asked me in 2022, do I really study metastatic disease? I would have said no. And now we've kind of got new tools that have got me really excited about what we can do in this space to learn about what makes metastatic ILC and the unique ways it presents in patients different. So again, welcome for joining us both in person and online. I'll hand it off to Tracy and Donna to talk about Front Range ILC and what they took away from the 2024 ILC Symposium. Thank you again for being here, everyone.
Hi, everybody, and thanks for being here. And we just also wanted to give a quick shout out to LBCA, and they're bringing us all together to celebrate the first Lobular Breast Cancer Awareness Day. Unfortunately, our proclamation request to Governor Polis went unanswered, and we were not one of the lucky states that got an official proclamation to hang on the wall, but we are unofficially proclaiming this as Lobular Breast Cancer Awareness Day here in Colorado, and across the world. So we really appreciate the work that LBCA has done for us. Just really quickly, for those of you that don't know me or Donna, I'm Tracy. I was diagnosed with lobular breast cancer in 2021. So I'm still fairly recent on my journey. I had a double mastectomy. I had my ovaries removed. I've been enjoying the surgical menopause for 3 years, been on letrozole for 3 years, and on abemaciclib for 18 months. And when I was diagnosed, abemaciclib wasn't even approved. So I think we're making some progress with ILC. In the meantime, I had just been busy practicing emergency medicine, not paying any attention to oncology at all until I was diagnosed, and then kind of threw myself into the world of advocacy, as well as integrative oncology. And I have a private practice doing that now. I'm a founding member of the Front Range ILC. I'm the former chair of the board of directors of the Lobular Breast Cancer Alliance. And I'm currently pretty active in grant reviews for the Department of Defense and for the Komen Grant Review Program.
Good morning, everyone. I'm Donna. My kind of history, I have a longer relationship with breast cancer than Tracy. I was first diagnosed when I was 39, which has its own challenges when you're talking to doctors. But at that time, they found both lobular and ductal, and the short version of it is that they said, well, we can do a single mastectomy and then you're good. And I was just about to move to Colorado. So I moved to Colorado, established myself with an oncologist, and he was like, great, nice to meet you. Hope I never see you again. He literally said that. And then I had a fantastic 10 years with no breast cancer in my life. And I was busy with my career. And then in 2019, I had a recurrence, which turns out to be invasive lobular breast cancer. And many of you know that lobular breast cancer tends to form in strings or sheets, but I actually had a mass that was visible on ultrasound, and it was right in the location where my previous breast cancer was. So that actually turned out to be great because they were able to identify it and treat it. And so I just celebrated 5 years of, I should say, acknowledged 5 years of living with breast cancer, but I actually turned 55 this year. So I had a party this summer that was my 55-5, and we raised money for breast cancer research through that. Tracy and I have been working to establish the Front Range ILC, but really we are just, what do I want to say, facilitators, and that really this group is all of ours. And so we're really hoping to hear from you in terms of what kind of things you would like to see or do. And with my stage 4 diagnosis, it's in everywhere in my bones, and I'm happy to say that at the moment I'm stable and not in any pain, and I'm just about ready to ditch the cane. So that's good. And if any of you are on Instagram, I have an Instagram that's dedicated only to stage 4 breast cancer. So I'd be happy to connect with you there. Okay. So the Front Range ILC group, we really wanted to kind of develop a space where we could interact with each other and with Matt's lab and really make sure everybody kind of understands what ILC is and what the science is around it, and then also to support each other. So it's really difficult to have breast cancer and to go through it. And then especially when you hear that you have lobular and the treatment is the same as ductal, it becomes very worrisome. So as Matt said, we've had 3 other events that were sponsored by his lab. We have had a couple of meetups this fall, and we're hoping to hear from you all in terms of if you want to meet up in person or if you want to do virtual meetups. And this is growing. We're happy that it's growing, but I always feel a little bit melancholy whenever there's a new request, because we're happy that you found us, but sorry that you're here.
So largely we've relied on communicating through our Facebook group. It's open to local patients, caregivers. We're happy to have clinicians that want to learn about lobular from a patient perspective. We thought, once as the chair of the LBCA meeting Matt and knowing there was a local lab in my backyard that was doing research on my rare cancer, I felt like it was a really unique opportunity to partner with a basic science researcher, which most breast cancer advocates really don't have the opportunity to do. And so part of the impetus for creating this group and, getting everybody involved is to both keep patients abreast of kind of the research, abreast is probably not the best choice of words, but to keep patients involved in what's going on, and to kind of show them the places where these discoveries are being made and to give them the opportunity to support Dr. Sikora's work as advocates, and in other ways, and as well as to support each other. So we also hope to liaison with our local LBCA advocacy groups, and really our goal is to share awareness, to give patients the awareness and education and the tools that they need to advocate for themselves. It's not a rare disease, it's not ductal, and it deserves kind of special treatment. And with that in mind, there is a Lobular Symposium that took place for the past 2 years, is now going to be not, I hear, in 25, but in 26. And Donna and I both had the privilege to attend this year. So we're just going to give some perspective on our experience and our takeaways.
One thing that I want to just back up with, so one of the things that Dr. Sikora mentioned was the Department of Defense and breast cancer. And if you're like me, you may be like scratching your head like, Department of Defense is funding. They're actually one of the biggest funders of breast cancer research. It turns out that a lot of active military and veterans have a higher incidence of breast cancer than the general population. And so they have dedicated a lot of money to breast cancer research, which is great because the Department of Defense budget is pretty big. Okay, going back to the conference, I want to actually thank LBCA for a travel grant, which allowed me to go to Belgium to participate in the symposium. I'm trained as a physical scientist, so I've been to a lot of science conferences, and I walked away from this one just completely impressed. I didn't have the opportunity to go to last year's conference. And so the thing that I wanted to really share as a takeaway is that there were just over 180 people at the conference, and that included patient advocates like Tracy and myself. There were probably, I don't know, 20 advocates, 30? Yeah, at least 30. There were clinicians, so people that are actually working with patients, and academic researchers. And all 3 groups were talking with each other. They had panels that had all 3 groups represented. This is highly unusual. So if you're not familiar with science, like it generally doesn't work that way. And so this is really encouraging, because the way that we can make more progress is by sharing experiences and information. The other thing that I will say is that the entire group that was there were highly motivated and passionate and really want to do good work, and they want to hear from patients. And so that also was really refreshing. And then there's Matt just showed us an example of progress being made. This image that's on the lower left is, sorry for it being grainy, but it goes from, on the very left is 1941, and on the right, 2024, so this year. And this chart shows the number of academic publications related to lobular breast cancer. And so you can see that it's just increasing significantly from probably zero in 1941. So that's really encouraging that there's so much activity happening. And then the picture that's on the right is the main room where we had all the sessions. And so it was set up so that a single speaker could be at a podium like this, but it was really inviting that they had a lot of chairs and tables kind of in the front for the panels. And so it didn't feel like your typical stuffy scientific conference where people are behind, where there's something between you and the presenter. And now Tracy's gonna give our highlights. I'm gonna go sit down. Is that okay?
So I had the privilege of going to the Pittsburgh conference last year, and I'm a physician. I've been to many scientific conferences, and I have never been to a scientific conference that has almost equal numbers of patient advocates, clinicians, and researchers as the International Lobular Symposium. It's quite rare, and it was quite inspiring as a patient advocate to know that my opinion and my presence was not only wanted, but very much sought after. And the conference in Leuven was largely helped to be planned by patient advocates as well as scientists. So, and that's just absolutely almost unheard of in other types of breast cancer meetings, in oncology, in the House of Medicine in general. So we are, excuse me, we are uniquely lucky in the lobular community to have such a dedicated and highly passionate group of glob mobbers that care so much about lobular research. And to that point, day one of the conference was almost entirely for the advocates. And so we got to take a tour of the University of Leuven Pathology Lab. We got to see exactly what happens when you have a tumor sample taken, the sort of journey of the sample and how it gets through the pathology lab. We also learned about their clinic, which they have there, which is a specialty lobular breast cancer clinic. I took away from that very specifically that their imaging test of choice is whole body diffusion weighted MRI, not FDG, PET, or CT, which it is here. And as a patient and advocate who wants the very best, I was curious to learn more about their opinions on why that was the imaging test of choice. So in the, excuse me, sorry. So in the afternoon, of the first day, we had 3 separate sessions, again, all driven by patient advocates, talking about different challenges for the ILC community and patients with ILC, one of which is imaging. So from screening all the way through recurrence, all the way through tracking of disease progression, we know that traditional imaging for ILC just often doesn't cut it. So I had a 3.5 centimeter tumor of an easily palpable mass. It was completely invisible on a mammogram. 5 years in a row, completely invisible on a mammogram. Even with biopsy clips in it, completely invisible on a mammogram. And so that was the screening that failed. And for lots of various reasons, other traditional imaging doesn't work. So it turns out estrogen receptor positive tumors aren't always as glucose avid or sugar hungry as other tumors. And so FDG PETs that use sugar as a tracer don't always find it. And so this is these results here are on distant metastatic disease, talking about the sensitivity and specificity of whole body MRI versus CT scan. And you can see there's quite a bit of difference between 98% versus 63% sensitivity for whole body MRI. Try getting an insurance company in the US to approve a whole body MRI for you. It will be almost impossible to get. So day 2 then was kind of delving more into the science, some things that I took away from it. In addition to the sort of ECADHERE CDH1 mutation that we all are mostly familiar with in ILC, quite a few of us in the metastatic setting will have PIK3CA mutations, and that was the second most common one that was found. And I do know that there are some exciting advances in targeting that as a potential therapy, as well as promising for HER2 mutants or HER2 low tumors as well. Again, the pathological diagnosis always seems to be tricky in getting all the pathologists to agree on that. And so there were sessions on how we're moving towards international standardization of diagnosis. Another challenge, particularly in the metastatic setting, is that often lobular tumors are not measurable by certain research criteria called the RECIST criteria, which tends to look for volume of tumor and then treatment and then less volume of tumor. And if your tumor doesn't grow in a typical way that you can measure its volume, then you're often excluded from those research projects. And so trying to, or from those clinical trials. And so trying to change the way we define and image lobular breast cancer so that we can apply it in more clinical trials. Then the session on dormancy was absolutely fascinating. Basically, if we think there are little cells floating around that are asleep for 10, 15 years, and then all of a sudden they decide to wake up, why does that happen? And is there anything we can do in that setting to just keep them sleeping, which frankly would be fine with me. I don't care if they sleep. I just don't want them to wake up. And then finally, on day 3, 2 super fascinating topics, the tumor microenvironment and then lifestyle interventions in breast cancer. And there's lots and lots of really interesting work being done on what types of cells live around the lobular cancer cells and help them grow and support their growth. And maybe the way to prevent them from growing is to actually intervene on the cells around them that support them instead of the lobular cells themselves. And the pattern of cells growing around lobular tumors is quite distinct compared to other breast cancers. And so what is it about those cells and the particular type of cells that either protects them or hides them from our own immune system or causes them to grow and support them? And is that something we could use as a way to modulate therapy? And then finally, when I was diagnosed and I was spiraling out of control about, what did I do wrong in my life and how did this happen and why me? And I asked my oncologist what I could do to prevent a recurrence, knowing that, life is a crapshoot in many ways. She said to me, you can exercise. And so I've really been interested in the link between things that I could control and making my cancer not come back. And in particular, there have been some interesting studies on body mass index, which isn't an ideal measure of weight, but an adipose tissue and whether lifestyle interventions can actually help prevent recurrence. And there are these 3 separate curves of recurrence that you see in folks with different body mass index. And my own particular theory that's based on nothing other than these research, basic research is that as we exercise, the changes that happen in our body as we flush out lactic acid, as we keep our muscles and our cells active, also keeps our microenvironment hostile to cancer. I'm hoping that someday somebody will prove that. Right now, I don't have any proof for it, but there is this neat trial going on right now with exercise and an aromatase inhibitor where they're looking at recurrence rates as well in Belgium. So those were kind of the geeky science things that I took away from it. But I truly just want to say thanks so much to Christine Desmet, who largely planned it, and to all the advocates who were involved in planning it, because it was truly a collaborative conference. And I highly recommend, if you have the opportunity to attend in 2026, which I hear is going to be in San Francisco, that you should all go.
All right. Thank you so much for that. Did anybody have any questions about the conference, the science that was presented?
Quick question as regards to BMI, exercise, and obesity and breast cancer. when you were there in Europe, are you seeing different treatments as far as weight management goes? Because I know that on aromatase inhibitors, it's difficult to lose weight. Plus you're in supermenopause. Is there anything looking at medications such as Ozempic or Wegovy, anything to help these women? Because speaking from experience, it's been very difficult for me to lose weight despite being athletic and fit and all of the things.
So all of the research was on behavioral and lifestyle interventions, but they have very aggressive patient support programs over there where like an exercise physiologist is part of your treatment team and a nutritionist is part of your treatment team. And so they really try to set you up for success without medications. I didn't see any specific research on pharmacology, but there was lots of research on behavioral and lifestyle interventions in keeping patients physically fit and healthy and active. Because yeah, it has lots of effects both on how well you can tolerate treatment and for long-term survival. So yeah, there's lots of it, but they have a lot of the supports built into their patient care teams that I think we're sometimes lacking here.
I'll just add one thing. If you are on Facebook, there is a group for breast cancer that's called something like Trials and Innovative Treatments. And that is, and if it's not right, somebody can reach out to me and get you the right name. But there's discussions. I know there's been a discussion about Ozempic, but that whole site is really, that group is really focused on research-based, activities. And so that might be a good place to look or to even ask a question.
Thank you.
All right. Now I'm really excited to share a little bit more about research updates, some of the big things that we've done over the last year. And I'll focus mostly on kind of the most translational aspects or the things that I think will be most immediately impactful to the folks in this room. Please stop me if you have questions. I'd love to explain the details and kind of make sure I'm not going over anybody's head with the science. So I alluded to earlier kind of two areas I'll focus on today. One being from the event, our first event back in June 22, where I alluded to this idea that we'd identified that lobular cancer cells are potentially bad at DNA repair. And I'll go more into this and a potential role for a class of drugs that are already FDA approved in breast cancer called PARP inhibitors. So I'll talk more about that. And as I mentioned, I'll talk more about where my lab is making new inroads, I think, or in building new models to study metastatic lobular breast cancer, why it goes to weird places, why it behaves differently, and how we have new opportunities to study this process. Before I go into the science, I'm going to focus quite a bit today on what we're learning from mouse models of lobular breast cancer. I want to talk a little bit about why we use animals as models and that we take that research very seriously and not lightly that we're doing this. So this is going to be a couple figures from a commentary I did with Becca Riggins at Georgetown a couple years ago now. The state of laboratory models of lobular breast cancer has changed dramatically over the last few years. We have a lot more options now. And in general, you could break it down to this idea that we have cell lines, we have organoids, which are chunks of patient tumors that are growing in culture, and we have mouse models of lobular breast cancer. They all have their place. There are different aspects of lobular biology, their growth features, and their genetics that are recapitulated by each of these three model types. They're not any one of them is perfect. The adage we use in science is that all models are wrong. It's just picking the one that suits the question you're asking best at any given time. It's also, when you're thinking about one size fits all, there's a big cost to be gained as far as how complex a model is, how much it costs, how much time and effort it costs, and things that you can actually learn from the disease. On the simplest end of the spectrum are these cell line models, these immortalized cells that have been growing in culture for decades. They're simple, they're relatively inexpensive, but we can learn a lot from them. There's a lot of ILC biology that is recapitulated. But as you can imagine, cells growing in a petri dish cannot recapitulate things like the tumor microenvironment that Tracy talked about. So there's a spectrum of which models we choose based on the question we're asking. So we try to use the most appropriate model given all of these different criteria and what we're trying to accomplish. This is a statue that I think a lot of us, it means a lot to. It's a monument to the laboratory mouse in the Russian Academy of Sciences. My lab does not take lightly what we ask of our mirroring colleagues. So when I show you this data, please know that we don't take these studies lightly when we do these things. So something my lab has been interested in for a number of years now is identifying what makes high-risk ILC unique and how we can learn more about it and ultimately target it. A few years back, the Cancer Genome Atlas, which was a huge NCI effort to characterize like every cancer ever, in lobular breast cancer identified one particular subgroup of lobular tumors, which they term proliferative, that had the shortest disease-free and overall survival. And those are those green lines here. So those would be arguably denoted then as high-risk ILC. In the TCGA data, it was not abundantly clear what made this high-risk group unique, but what stood out to us was this DNA damage response feature. And this is a measure of proteins in the cell that are related to the cell's ability to repair damage to their DNA. And this so-called proliferative subtype had very high levels of this DNA damage response signature. At the same time in my lab, I showed you this image earlier, but we started to learn that these lobular cells in culture aren't repairing their DNA the same way that ductal cancer cells are, or other breast cancers of no special type. These images are fluorescent microscopy images, so we're zoomed way in onto these breast cancer cells that are growing in plastic. And the blue is actually the DNA or the nuclei of the cell. And we've hit these cells with a chemotherapy, and then we look at these red and green signals. Each of these represent two parts of the cell's machinery that it needs to fix DNA. So in a ductal cancer cell, all of these little puncta, these little Christmas light looking dots, are actually physically these two parts of the machinery getting together at an individual site where DNA is broken to fix it. So the ductal cancer cells are quite good at it. They get the machinery to the spot that's broken and they fix it. Eventually these, they're called foci, will resolve, they'll go away, which means the DNA is fixed. The lobular cancer cells are really bad at this. You can kind of see that some of the cells do have some of those foci back there, but most of the cells that we see have this red haze. And we think it's because they're turning on the response. They recognize that something is broken, but they're really bad at getting the machinery to where it needs to be. Something is dysfunctional about their ability to repair DNA. Now, this is not without precedent in breast cancer. Essentially all BRCA1 and BRCA2 mutations, those lead to breast cancer because it leads to a dysfunction in DNA repair. So is this phenotype in lobular cells related to mutations or loss of function in the genes BRCA1 and BRCA2? The answer to that is largely no. BRCA mutations are actually fairly uncommon in ILC. Other mutations in the spectrum, notably BRCA1 and POLB2, are actually strongly depleted among ILC. And none of the cells that we have in culture have those mutations. So kind of the low-hanging fruit that would explain this observation is not the case. We also know, though, from TCGA and other data sets that lobular tumors don't have any of what we call genomic scars of BRCA-ness or this BRCA mutation. These mutations in tumors actually lead to measurable scarring in the genome where DNA gets broken and can't get fixed properly and the cell has to kind of Frankenstein it back together. In ductal cancers, we can often see scarring of that BRCA-ness phenotype, but lobular tumors show none of that. So there's not an obvious reason that these cells would be so bad at repairing their DNA. More recently, we've actually done kind of functional assays to see how cells repair their DNA. This part is these images here are the very initiation of DNA repair. The cells have recognized the damage and are trying to fix it. And this is a measure of how the cells actually fix it. This is a really cool experiment, and I wish you could see how much work goes into like a few bars, but I'm sure Joe has something to say about that in the back. Anyway, this experiment measures two major repair pathways, homologous recombination and non-homologous end joining. The important part is that this HR pathway is what the BRCA proteins use. They focus on that particular repair pathway, which is very high fidelity. It repairs breaks back to what was there in the first place. In this experiment, we have a couple of models of ductal cancer. They are happy to perform either repair pathway, which is totally normal. Cells will pick one or the other based on other facets of where they are in their lifespan. But we found that lobular cells apparently have little to no interest in using this HR pathway, this homologous recombination that is otherwise downstream of those BRCA proteins. So this has had us thinking for a while now whether or not ILC actually have a unique and previously unappreciated form of BRCAness that is not tied to what we currently know about BRCA mutations. So why is that important, BRCAness, and thinking about that? It is tightly tied to the use of these drugs called PARP inhibitors in the clinic. The way PARP inhibitors work is that in our DNA double helix, cells will encounter a single strand break often. This can be just through to normal processes, the cell dividing, oxidative stress related to metabolism. This is a routine thing that happens in all of our cells all the time. There's a class of proteins called PARPs that are largely responsible for coordinating repair of those single strand breaks. In a normal setting, that gets repaired. In cells that are otherwise competent to do these repair pathways like HR, the use of PARP inhibitors will cause that single strand break to ultimately turn into a double strand break. So in that case, the long continuous strand of DNA gets physically broken, and that's very bad for a cell. A normal cell will do everything it can to use, in particular, homologous recombination to fix that. So that cell is alive. However, in cells in particular that have mutations in the BRCA genes, they cannot use this repair pathway that's homologous recombination. They can't repair that double strand break and the cell dies. That paradigm right there is central to why patients who have germline or hereditary mutations in BRCA, their tumors have lost this repair pathway and their tumors are then highly sensitive to PARP inhibitors. But as I mentioned earlier, though, lobular tumors are depleted in BRCA mutations and have no other genomic features of this BRCAness phenotype. So patients with ILC are actually rarely eligible for PARP inhibitors. By all our current measures, none of you are likely eligible for that drug. However, with what I just showed you a moment ago with those pretty pictures and what we're learning about DNA repair in lobular cells, are those dysfunctions in DNA repair, or do they actually cause a novel form of PARP inhibitor sensitivity? So this is where we've done a lot of work with our cell lines and culture in the lab, but ultimately to move this forward, we need to test this in vivo tumors. So what we actually do routinely is to take fragments of patient tumors or those cell lines that have been growing in culture. We can implant them into or at the mouse mammary gland, which is our effort to get as close to a human breast as we can. The tumor will grow, and then we can treat the mouse with a given cancer drug. And then we wait for the response, measure these tumors, and we can use this to study how the tumor responds and how it doesn't respond to therapy. The drug we're using as far as testing PARP inhibitors is Pfizer's talazoparib or Talzenna, and we're using this as a model combining with aromatase inhibitors. The actual model in the lab isn't using the drug entirely. We're changing estrogen levels, and I'd be happy to talk about that later. But suffice to say, we're testing PARP inhibitor plus or minus aromatase inhibitor as a model for advanced disease. So this is just one example of what these studies look like. In this case, what I'm showing you here is 4 groups of tumor experiments. Each of these represents 5 hosts that have tumors on each side of their body. So you're seeing individual growth lines from 10 different tumors treated with these different therapies. Then the solid lines represent the average of the group. This y-axis is the actual tumor volume. We just measure that on the outside using calipers just to measure the outside of the tumor. And then here is weeks after the mice were inoculated with the breast cancer. What I will highlight here too is that lobular cancer grows really slowly in mice, which is a pain in the butt from a technical standpoint, but it probably actually much more closely mimics what lobular cancer does in patients. Most labs, when they're studying breast cancer therapeutics, they're done at like 4 weeks, and we're going out to 28, 32 weeks. So the folks in the lab are very dedicated to these studies, but I think it actually is a better model in the end. But what we see here is that in these 4 groups, so we have mice where we just gave them a vehicle control, or we gave them a mimic of how we treated the rest of the mice. A group of mice received talazoparib. They actually received this orally. Ask Cameron and Joe how that actually works. We use our model of aromatase inhibitor, and then we have this group that gets the combination therapy. These mice were actually treated for a very short treatment window, only 6 weeks. In the clinic, a patient would stay on this as long as their tumor does not recur. So this could be for a very long treatment window, but we actually intentionally wanted to see what happened at the completion of therapy, whether the tumor could come back or not. In the black line are the untreated tumors. So these tumors just continue to grow happily, and eventually the mice reach humane endpoints. And then talazoparib alone didn't really do a whole lot. In this model, this is one of our cell lines. Aromatase inhibitors do strongly suppress growth during that 6-week treatment window. At the end of treatment, though, we actually give them estrogen back as if the aromatase inhibitors were stopped, and these tumors come roaring back as if nothing ever happened. So they're very estrogen dependent, but they never really regressed fully. They were there, and as soon as they had their their growth signal back, they just roared right back. I will note that these mice, since we're using human models, they're all immune compromised, so there's whole parts of tumor clearance that we can't measure. So there's a little bit different with how response works here. But what was really encouraging here is that the combination of aromatase inhibitor plus talazoparib, not only did we get a stronger inhibition and reduction in tumor size on treatment, but these tumors never really got it together after treatment was completed. We're about 3 months after completing therapy, and these tumors never really figured out how to outgrow again. They do get bigger, and eventually we have to euthanize the mice for humane endpoints. But clearly, we're having a long-term effect on the tumors just by adding this PARP inhibitor, which is really exciting from a treatment standpoint. And then beyond using cell lines, probably what's considered now really the gold standard for looking at these tumor responses in these mouse models are actually using what's called patient-derived xenografts. And these are fragments of patient tumors that have never been on plastic. They've never been in a Petri dish. They originally went right from a patient's surgery right into a mouse. So they arguably better represent what's going on in a patient since the cells never had to adapt to grow on plastic. Same idea with treatment study. We actually, for each treatment, had 2 groups of mice, which is why you have 2 lines. So seeing the lines line up is just very gratifying from a statistical reproducibility standpoint. And then this is a little different. We're looking at a log scale of change in tumor size over time. In this model, again, the control tumors grew very quickly. The talazoparib alone didn't do much of anything. This model is also a little different because it has an ER mutation, which we know is a way that tumors resist aromatase inhibitors. And you can see that the tumors, they didn't keep on growing very quickly when we used the aromatase inhibitor, but a lot of them kind of hung around full size, and a lot of them started to regrow by the end of, in this case, a 10-week treatment period. The addition of talazoparib, though, much more strongly suppressed tumor growth. A lot of these tumors are bouncing around our ability to even measure them in the mice. Down here where they're called non-palpable, we can't even find them in the mice. And if we look at actual treatment start and end, in this case, I'm showing you the aromatase inhibitor compared to the combination and the tumor size at start and at end. On aromatase inhibitor alone, about half of the tumors were either stable or actually started to outgrow on the treatment frame, where about half of the tumors responded. Them again, we couldn't even find when we're trying to palpate them in the mouse mammary gland. So we're really excited about the potential for exploiting what looks like a defect in DNA repair with a drug that patients already get. Just by current definitions, patients with ILC aren't going to get them, and we need to fill in that gap. So this is, I'm really excited about this leap. We're going from these immunofluorescent images in the lab that mean nothing for patient care to plausibly something that is a tractable therapeutic approach that we could advance in clinical trials. I'm really excited about a partnership that has developed over the last year or so with on campus, the Women's Cancer Developmental Therapeutics Program. Jen Diamond is a physician scientist in medical oncology and co-director of this program, WIC-DT, who has essentially signed on as kind of my clinical partner in this space. And we are starting to design and propose clinical trials in this space. We're not there yet. We have a few more hurdles yet to go, but I am really thrilled about how the lab has moved this forward from little pretty dots to plausibly impacting patient care. So stay tuned. We're trying to move this forward. Still a lot of hurdles to go, but we're as close as we've ever been to actually moving this into care. All right. That's our therapeutic space. And now I'll talk a little about metastasis. This is much more basic, much more modeling and trying to understand how this disease functions and what opportunities we have for more experiments. So normally when in those experiments I just described, when we're thinking about growing breast tumors in mouse models, this is a diagram of where the mammary glands are in mice. Usually we're implanting tumors into the fat pad or the fat surrounding the mammary gland around these 2 nipples here. We're not usually able to go into the breast structure. We're just going next to it for practical reasons, but it does provide a relevant microenvironment. There's been new advancements, though, and new methods in actually putting the cancer cells directly into the mammary structure in these mice. And rather than putting it in the fat pad where we would normally put it in this space nearby, we can actually inject cancer cells directly into the mammary structure. And in this case, we add a dye, and you can see that the dye, with in this case full of cancer cells, fills up the entire mammary structure. So now the cancer cells are growing in the mammary duct, which is where they arise in the first place. And when that happens, the cancer cells actually have an opportunity to invade the normal mammary gland, and they have to go through the entire full metastatic cascade that a patient's cancer cell has to go through. It starts in that normal encapsulated mammary structure that is the mammary duct, and then eventually has to escape to invade into metastatic sites. This is arguably much more relevant model for breast cancer metastasis, since otherwise, if you're injecting into the fat pad nearby, the cells are starting out in this space, and they can kind of skip this entire process. And they're also just, they're provided a different microenvironment that isn't quite the one that they ideally would have compared to a patient tumor. When we actually do this, the tumors metastasize to all of the places that a patient may experience, including ILC-specific sites like the ovary. This is an example of a cross-section of a mouse ovary. What's really cool, what we're seeing is that these are not just little micrometastases, which often plague some of these models. It never gets to an overt metastasis that you can actually really study. But in this lobular model, when we put these lobular cells into this intraductal setting, this is a cross-section of a mouse's ovary, and these giant nodules here are all lobular cancer cells. This is an example of using estrogen receptor staining to kind of highlight the human ILC cells in the mouse ovary, and this is a massive tumor for a mouse in the ovary. So it's really exciting mechanistically that it can go there. The other really interesting site is in the brain. Lobular tumors are unique in that they go to the membrane surrounding the brain called the leptomeninges, and there really are no good models for spontaneous metastases of an estrogen receptor positive disease to the brain. I'm going to show you a black and white image of a mouse brain. If that sounds unappealing, look away for a moment. But these tumor cells have been engineered. They also emit light. We can actually track them in various imaging tools, and if you see light coming from a tissue, it is the cancer cells. Here comes the black and white mouse brain. So this is actually an intact mouse brain, and we're looking for light emitted from cells. And there are these two screaming puncta of light in these tumors. And if I were to actually show you the intact picture, these are two huge brain metastases. And this is really exciting because it happened spontaneously. These cells had to go from the mammary gland, they got into the bloodstream, and they made their way to the brain. I'm sorry, this is getting washed out from the sunshine, but these cells are actually, this is a cross-section of the mouse's brain, and all of these cells that look weird are the cancer cells, and they are actually surrounding the membrane structures. These are the ventricles in the brain, and this is, sorry, I can barely see it now with this. Where am I pointing? This is the outside membrane of the brain, the leptomeninges, and you can see the cancer cells colonizing there. Very bad for the mouse, but very exciting for us that we can actually model plausibly what's happening in patients that is unique to lobular tumors. Yes? I'll repeat the question. The question was whether it's staying outside the actual brain. Hard to tell yet. Right now, it comes down to dissection techniques and how we actually physically can look at this. Joe back there can talk more about, can you say anything about how we actually physically see where they're at? Dr. Joe Sotnick leads these experiments in my lab.
We are able to see that there is some invasion into what more typically is gray matter, white matter, but it seems to be that there is a bigger growth in this space and fluid that surrounds the brain, which, as Matt said, kind of recapitulates more of what we see with ILC than what is seen in other breast cancer metastases to the brain.
Thank you. Oh, yeah.
Thank you. Quick question. Earlier, you had stated that it gets into the bloodstream. Are you seeing that ILC is traveling via bloodstream as well as lymph, or is it both, or?
In these models, we don't know yet. Right now, we're kind of operating kind of in presumptive models based on other breast cancers. They probably have to go through the bloodstream, but we would have opportunities to start testing whether it goes through the lymph or whether it goes through the blood. And we're starting to get kind of the experiments in place to ask those questions. But it's a really good question.
Thank you.
Yeah, it's worth considering. A lot of the models we have for breast cancer metastasis overall all hinge around the cell adhesion protein E-cadherin and how that protein allows cells to grab onto the microenvironment and pull themselves in, but lobular cells lack ecadherin. So all of the models we currently have for breast cancer metastasis essentially don't apply to lobular. So we're trying to reinvent now how we ask these questions using models like this. Some of the, I alluded to some grant proposals with colleagues at the Hutch, and we're starting to ask, what are lobular tumors using to actually grab onto these other sites, the ovary, the brain, pull themselves in and survive? So with these models, we can ask these questions now to fill those gaps like that and where they're going. No, please. This is why we're doing this.
I'm nerding out on all of this, excuse me. So with the brain, are you seeing any different behaviors with the mice when they do have the metastasis? Sorry.
No, so these are actually symptomatic and lethal metastases, which is the worst for the mice. But probably the coolest part about this model is that, again, these are not just little cells that are hanging out. These mice are dying of brain mets. So they get central nervous symptoms, they get head tilts, they spin. They have a bad time, unfortunately.
Okay, thank you.
Yeah, it's tough. And like I said, we don't take these experiments lightly, but with the changes to the microenvironment, we simply cannot do this outside of these animal models.
With the brain, are you able to, the blood-brain barrier is what I'm talking about here.
Mhm.
So would the medication be more effective if it's administered through the cerebral spinal fluid versus vascular? I'm just.
Yeah. No, so there are absolutely trials with other therapeutics to try to get over the blood-brain barrier. Talazoparib, as an example, is supposed to cross the blood-brain barrier. It may or may not do so super well. So we are cognizant of that. And that's part of what we can do in this model is when mice get these metastases, we can treat them kind of past a point when we think they've already got metastatic establishment. We can wait that long to treat them and then follow metastatic outgrowth from there to see if we're differentially impacting, say, bone mets versus brain mets. Joe, do you have anything to? I got it? Okay. Joe makes sure I don't say anything stupid when I'm talking about metastatic research. He's the expert. Yeah, so we have a lot of opportunities to ask those questions in this space. Yeah. So in addition to the ovarian and the brain mets that are essentially lobular specific phenotypes, these mice do also get micrometastases to the lung. They get GI mets, which are unique to lobular patients, and they do get bone mets. And actually, we just got some recent internal pilot funding. Joe's going to do some mechanistic work to understand what these cells are doing in the bone in particular. And what we're also seeing is that, again, it takes a long time. But ultimately, all of these mice do get all of these metastases. So we have opportunities to study this. It takes a long time for all of these mets to show up. We're talking, over half a year at this point. But it does afford us really cool windows of opportunity to intervene and to identify places where travel through the bloodstream happens, travel through the lymph system. So we have time to look at this and understand the process, as opposed to a lot of lab models, you're injecting cells into the bloodstream, metastases happen instantly, and you have no mechanistic window. Yeah.
So the uterus metastasis, that is something that I've heard as only if you take like tamoxifen, not if you are on aromatase inhibitor, right? Is that something new that we are kind of?
I'm sorry, which
identified?
metastasis?
Uterus.
So as far as I understand, the metastases are not explicitly linked to tamoxifen. Tamoxifen itself does have a small but real increased risk of uterine cancer. I have not heard a concrete link for lobular metastasis to the uterus, though. For tamoxifen, obviously it does go there normally, but I'm not aware that it's linked to tamoxifen explicitly.
Oh no, what I meant is like if someone is on aromatase inhibitor, if you have lobular, is there still a risk of uterine metastasis based on like what you're sharing here?
We haven't tested how different therapies like that impact metastatic spread yet. Now that we know that these models do this and we understand the timing, we can start intervening at different points
Okay.
to start asking those exact questions. Yeah,
Thank you.
and that's the plan. Okay, so just to wrap up this section, this is where we started as far as kind of observations and where the lab is going. Over the last couple of years, we've taken a kind of side comment about a potential role for PARP inhibitors, and we're testing this in relevant preclinical models that can help us advance this into potentially testing in patients. And we're going from kind of being aware of metastatic research to having bona fide efforts in this space, which with really new opportunities in models to understand how this cancer spreads and how it gets to different places differently than other breast cancers and how we can intervene. With that, any other questions? Anybody online?
This is Karen with a question.
Oh, hi, Karen.
Hi. It on your previous slide, it looks like the lung is the first place that this is traveling to. The brain and that you're studying. Would it be more useful to look at because it reacts faster, the lung tumor? Would it be better to work with that instead of the brain?
That's a really good question, Karen. So the time points of spread are a little biased because with our current imaging modes, we can't see the METs when we look at a live intact mouse. We actually have to go looking in the tissues for those light images. So we're a little biased by when we look as to when we identify metastases. We do have some thoughts with the different lobular models we have about potentially what order they're spreading in, but I'm not comfortable yet like picking one as like an initial site. It is worth noting that clinically, most patients would present with bone metastases first, and potentially bone only. So we do have some thoughts about whether that is a jumping board, and we can start to look at that in these mouse models. And the lung has been interesting because for some reason, these aren't really like heavily expanding in the lung yet. So we're not sure what these cells are doing in lung. The flip side though, you just reminded me, lobular patients clinically present with fairly few liver metastases. And that's one place that these cells aren't really going in the mice is the liver. So there is something about what these cells are recognizing, what they like and don't like in the microenvironment that is definitely important and that we're seeing recapitulated here. So we can definitely think more about that space, but yeah, still learning about the timing because that one's tough to measure. Joe's nodding, thank you.
Okay, I have a general question. So you talked about the fact, this is really exciting, by the way. You talked about the fact that you're using mice in the lab.
Mm-hmm.
Can you or Joe talk about like how many mice and like what's a reasonable number and like, do you find it in all of them or just one? Could you maybe talk a little bit about that aspect of the research?
Yeah, I'll give the concept level and then I'll give it back to Joe to talk about kind of the practical aspects. We try to use as few as possible, always. That's baked into all protocols that go through the university, and using animal models is you have to justify really using no more than absolutely necessary. So when we do this, we're usually only using 5 mice per any given treatment group, honestly, in part because you can house 5 female mice in one enclosure at a time. And that usually provides us a relevant statistical context to see changes in tumor growth. If we don't see it with each mouse has 2 tumors. If we don't see a change across 10 tumors and 5 mice and can't compare that, it's usually not worth chasing. So Joe, you want to talk about any other practicalities?
No, I think that's correct. We try and use the fewest possible for anything. And a lot of these experiments are also set up so we can get as much information as possible from any group. So it's not just when we do the treatment, we're also looking through as many of the organs and spaces and taking the tissues that we can. We process them in multiple ways so that we can look at all of the genetic information and the protein information and the slides that Matt showed with the histology on it. We can try and incorporate as much information as we can from a single one. But yeah, sometimes it's difficult. Like with these ones, we've been doing 5 mice per group. For the talazoparib ones that Matt showed earlier, that was 5 mice per group and 4 groups, so 20 mice. And it's mostly the time frame that's also a bit of an issue. So keeping mice that do not have immune systems around for 30+ weeks is a little difficult in its own right.
Yeah. I would also encourage you, Cameron's going to talk a little about her later, and she has a poster. You can ask her about what it's like to orally dose mice.
So, and I guess to follow up on the tumor microenvironment and how interesting that is, if you're using mice without immune systems or mice with mice immune systems and mice with mice tumor microenvironments, how do you think that affects the research and its applicability, knowing that only about 5% of actual therapeutics make it from mice models into successful human trials?
Yeah, as I mentioned before, all models are bad. It's just a matter of which one suits our needs the most. Right now, there are no immune competent models of lobular breast cancer that are ER positive. All of the mouse models that exist that focus on EC-adherent loss to model the disease they're all ER negative, or if they've been reported ER positive, it hasn't really been validated that they respond to hormone. So in my mind, they're probably modeling pleomorphic lobular for the most part, which is not uncommon for mouse models. They just don't, like a genetic mouse model of cancer doesn't usually make an ER positive disease. So we just don't have an immune competent one. And that is undoubtedly a problem. I guess the benefit for the PARP inhibitor studies we're doing is that an immune system should, in theory, enhance the response. Because PARP inhibitors, part of their mechanism is actually to make a more immunogenic environment. But the immune microenvironment in clinical lobular tumors is backwards from every other breast cancer. So we just don't have a way to ask that, frankly. There are ways to humanize the immune system in mice, but it would increase our experimental costs literally 10 to 15-fold. And it would be questionable how well it works. But you can actually ablate the bone marrow in a mouse and essentially give them human bone marrow. So they have a human immune system, but it's a lot. It's a lot. So again, it would be a guess. It would be too much guessing for our experiments and frankly, too costly to do right now. I think what we can do is get the best experiments we can with these immune compromised models and identify what the right translational model is. And the thing is, I think the big benefit is these drugs are already approved in breast cancer. So it's less of a leap than some other settings where we're like using a totally investigational agent. All models are bad. We're just picking the right one for any given circumstance.
So we have a couple questions from the chat.
Thank
So
you.
the first one is asking whether cell behavior is different for de novo metastatic lobular cancer.
Okay. That is a really good question. Another models are bad. All of the models we have come from metastatic cancer. It's not clear how much they behave like it. There's kind of a disconnect between how we model primary disease in the lab and and how we model metastasis. I could go on for a while, but getting primary tumors that are from the breast to actually grow in the lab is extraordinarily difficult for reasons we don't really understand. Essentially, any cell line you might see used in the lab came from usually a pleural effusion, the fluid that builds up in the abdomen. That's the most common site of where we get cells that happen to grow out nicely. The PDX I showed, the HCI 013, that I think actually came from a brain met. So the metastatic tumors are what grow out that we can study in the lab. Whether primary and metastatic study differently, we know more from the clinic, and the short answer is yes/probably. But I think as a field, we're still disentangling the contributions of the tumor and the microenvironment in those spaces. And for lobular, it's twice as hard because the tumor cells exist in those sheets. And so it's just physically harder to disentangle those. Technologies are advancing really fast to separate those in what's called spatial analyses, and we're getting there. I don't know if I actually answered the question, but that was my attempt. Let's see, thank you for the answer. I think I'm still wondering what research and treatment is most relevant to with de novo metastatic disease. Yeah, so as far as research, it is hard to disentangle the space. I think, and I would actually defer in part to the folks in the metastatic space, but how much you feel that research applies to your cancer and how you interpret research in those spaces. To me, as a researcher, in the lab models, we for the most part can't disentangle them. We're focusing on the tumor biology, and we're kind of intentionally putting a reductionist view on to say that the tumor is the tumor, not regardless of where it is, but we have to take a reductionist view. And then we use these type of models to start asking the more complicated questions. But as far, I guess, how you interpret research, would you care to comment on that? I'm handing off to Donna on this one.
Okay, I think that's a really good question. And like as far as, so I was not de novo, right? I had a recurrence. I mean, right now, all of the treatments are the same regardless of de novo versus recurrence. And so I think that, I mean, from my perspective, I think that it's an area that probably needs to be researched. If there's treatment differences, right? For de novo, that's I think what the question is. But I'm not aware of any current investigations that are looking at this problem, this challenge, I guess. So yeah, I have a non-answer for that. But I think that the standard of care for stage 4 largely behaves the same way whether you're de novo or not. So that's why I think there's probably not a lot of research into it right now, at least that I'm aware of.
I didn't talk much about it today, but we do have some projects that are thinking about how the selective pressure of therapy does change tumor biology, which would be the biggest thing between de novo and recurrent metastatic disease. And hopefully at some point we can start stratifying by that. We try to in terms of research, but in terms of what we actually have, I don't think we're there yet. For this next part, the folks in my lab, again, the people that actually do the work, I just beg for money. That's really my job. They are going to come up and talk to you a little bit about what they do in the lab, the projects that they focus on. We have a lot more going on than just even the 2 projects I showed you today, but I'm excited for you to meet them and hear about what's going on in the lab.
Hi, everyone. I'm Maggie. I am a postdoc in Matt's lab, and my project focuses on figuring out the beginning stages of ILC, so tumor initiation. So ILC is identified by ECADH loss or CDH1 loss. And interestingly, about 60 to 80% of the precursor LCIS also has ECADH loss, which suggests that normal healthy breast cells, one of the first changes of the breast cell to become ILC is ECADH loss. And so this is an early event that's happening to push cells towards an ILC state. So, So why do we care about ECADH loss? ECADH is a protein that's on the surface of a breast cell, and it's involved in communication between cells, and this communication coordinates movement of cells, growth, and survival. So in ILC, without ECADH, these cells are no longer able to coordinate movement, growth, and survival in the normal manner. So they can regulate it however they please. And so ECADH loss, we know, is involved with a lot of things that can push a cell towards becoming cancerous. However, we don't know exactly how this happens. And understanding exactly how it happens could help us be able to better predict which LCIS, for instance, could progress to ILC, and also help to predict or to develop better or more optimal ILC specific therapies. So to do this, most ILC research is done in ILC cell lines, which are fully invasive. However, as I said, ECADH loss is an early event in the progression of ILC, so we need different models. So the models that we have established to use are human mammary epithelial cells, which are primary cells, which means they're directly derived from patient tissues, from donor tissues, usually through reduction mammoplasty. And these cells allow us to look at CDH1 loss or ECADH loss in normal, healthy mammary cells, as well as in a kind of in between the healthy to LCIS state. And I currently have 4 different models that we're using, 2 derived from premenopausal and 2 derived from postmenopausal. So we can also ask the question of, or the importance or significance of menopause in this tumor initiation. And so a little bit of background on the breast tissue. This is the stuff that we have found so far using these models and looking at ECADH loss. So breast tissue is composed of several different types of cells, not just breast cells, but we have the cells that make up the milk ducts and lobules. We have fatty cells, and then there's fibrous network that connects them all together. The majority of breast cancer, including ILC, arise from the cells in the milk ducts. And these cells include two main cell types. So not all breast cells are the same. We have luminal cells, luminal cells, which respond to estrogens and anti-estrogens, AKA ER positive. And we have basal cells, which do not respond to hormones, and they are ER negative. The purpose for basal cells is to contract the milk ducts like during lactation. So interestingly, we have found that E-cadherin loss in the breast cells, in the HMECs, which include luminal and basal, has different effects depending on the type of breast cell. And so our current hypothesis here that I am excited to research more is that luminal cells, the E-cadherin loss in luminal cells actually reprograms the cell to a precancerous ILC-like phenotype or state. But this effect is not seen in the basal cells. So this is really exciting because if this is exciting because we can develop new biomarkers that show E-cadherin loss plus another characteristic that's only found in this subtype of breast cell could lead to an increased risk of developing ILC in patients diagnosed with lobular carcinoma in situ. And so with that, I'm really excited. I actually submitted a grant today to get funding to continue this research and improve prediction of progression to kind of prevent what happened with Donna, how, diagnosed with LCIS, get a single mastectomy, you're good to go. But now, hopefully, with this research, we'll be able to better predict which LCIS will progress.
Hello, everyone. My name is Carmen. I am a technician in the Sikora Lab. All right, today I'd like to introduce you to a really cool new technology that we just implemented in the Sikora Lab called single-cell RNA sequencing. The best way to describe RNA sequencing, in my head at least, is through looking at different things in space, this analogy. So in the first image, every dot that you see is a galaxy. From the image, we can tell that one galaxy might be brighter than another, and that would be because the stars in that galaxy are on average brighter than the stars in another galaxy. Although there are probably millions of stars in that photo, we can't actually distinguish anything about the individual stars or what they are doing differently from one another. So this same concept applies to bulk RNA sequencing. Bulk RNA sequencing has been kind of the go-to in the field for about the past 20 years. With bulk RNA sequencing, we take a tissue sample or a sample of thousands to millions of cells, and we are able to determine what they're doing on average. So we're able to draw some good conclusions from this, but we can't distinguish anything about what makes one cell different from another. This is where single-cell RNA sequencing is really powerful. It's more like looking at a single galaxy. And in this second photo, we can now appreciate the brightness of individual stars. If we wanted to, we could even group stars together into all of those that have a yellow hue or that are larger or smaller than others. We do the same thing with single-cell RNA sequencing. We look at each individual cell, in this analogy, each individual star, and we create a profile for it. Then we're able to group cells together into groups of cells that are acting similarly. So these bottom two images represent 40,000 ILC cells. With bulk RNA sequencing, we're looking at what they're doing on average. With single-cell RNA sequencing, we are able to break those 40,000 cells up into 12 distinct cellular states. So I like to think of cellular states as different phases that the cell goes through. Just like anything else, cells have a life cycle, and they have to move through each of these cellular states throughout their life. So by understanding each of the phases that cells are going through, we're hoping to increase our general knowledge of what ILC is doing. And we're also hoping that we can find new therapeutic targets that we would have otherwise missed by just looking at bulk RNA sequencing. Another goal of single-cell RNA sequencing is to develop biomarkers for therapeutic efficiency. So for example, if we know that the cell state that is represented here by cluster 1 is sensitive to a certain drug, and we also know that that cluster expresses a large amount of a given protein, then in a clinical setting, we could test a patient's tumor for that protein, and if it has a high level of that protein, we could make an educated guess that that patient's tumor would respond well to that therapeutic. So these are just a few of the goals that we have surrounding single-cell RNA sequencing, but it's a super powerful tool, and we are very excited to be using it. So one example of how I'm using it is to look at this protein MDC1. The Sikora Lab is very interested in MDC1 because it works with estrogen receptor to turn genes on and off in ILC. This bottom image, on the left, we're looking at lobular carcinoma. On the right, we're looking at ductal. The blue is the cell nucleus, which is where all of your DNA is stored. And the red puncta are the direct interaction between MDC1 and estrogen receptor. As you can tell, it's very obvious that the interaction between ER and MDC1 is unique to ILC. So this is why we're very interested in what MDC1 is doing in these cells. So I did single-cell RNA sequencing and was able to determine that cell states represented by clusters 2 and 3 are dependent on MDC1 regulation of their genes. This in itself was really exciting because, again, we would have missed this just using bulk RNA sequencing. So we are currently working to further characterize those 2 clusters to figure out what is different in them and how we may be able to therapeutically target that. So again, this is just one example of how we're using single-cell RNA sequencing, but we are really excited to have this tool, and I hope you think it's as cool as I think it is.
Hello, everybody. My name is Maddie Shackelford. I am a research assistant in Dr. Sikora's lab. And I just kind of want to give just kind of an overview of the things that I've been working on for a little while now. I am primarily working on 2 different projects, and these are kind of an extension of things that we've been working on in the lab for about the last 5-ish years. So my first main project here, oh, there we go. So as Carmen and both Dr. Sikora talked about a little bit earlier, is this ER-MDC1 construct that is unique to or complex that's unique to ILC. So we know that ER is binding to MDC1 and it's looking, activating genes. It's usually involved with recruiting to the DNA damage response. So then it goes and does that weird DNA damage repair that's unique to ILC versus IDC. But what we don't know is where exactly in MDC1 is the ER binding to cause this downstream effect. And so what I've been looking at or trying to find is, where is ER binding to? One way that we did this is by we had a couple hits that showed the unique potential sites for ER binding, and we made cell types that expressed each of those unique hits. So we essentially cut parts of this MDC1 construct here at those potential binding sites. And then we were able to use a method called immunoprecipitation to essentially isolate those unique cell types with just the MDC1 cut at that spot, pull it down and see, is ER there? That's currently ongoing, but we are pretty excited. We do have a couple potential hits, but ideally this will hopefully inform us as to not only where ER is with this within this MDC1 complex, but also how we can better target the ER MDC1 complex to potentially stop it from doing that damage repair and stopping the cells from surviving. The other project that I've been working on involves ER and WENT4. This is kind of a baby project for me. I've been working on it for a really long time. So we know that in ILC, WENT4 is weird. It does not behave how we would expect it to, and it leads to what we call atypical WENT signaling. Primarily what this is that we've been looking at how it is affecting metabolic remodeling, or this idea of causing ILC to draw from a different energy source than, for example, IDC. So usually we see that tumors like sugar, but we know that ILC does not like sugar. And we believe that it's possible WENT4 is what is driving that change with ILC to choose something other than sugar. So for example, fat. Very simple terms, which then is driving this cell survival. Ideally, what we would like to do is better understand how WENT4 is driving that metabolic change, so then we could ideally target that and stop this cell survival from happening, and therefore creating not only better biomarkers, but better diagnostic tools. Which is really, really exciting, frankly. But overall, yeah, WENT4 is my baby. Sorry, guys. I really enjoy talking about it. But yeah, I am going to pass it off to my colleague here, Sydney Robinson, who I believe will actually go a little bit more into about this WENT4.
Hi, everyone. My name is Sydney, and I work on WENT4 and specifically what WENT4 does with metabolism. So when we think about metabolism, we've all heard about metabolism in our day-to-day lives, but specifically it refers to the breakdown of different high energy molecules to create energy for the cell. And these molecules are in the forms of sugars or fats or also proteins, but for our purposes, we'll focus on sugars and fats. Sugars are also known as carbohydrates. Fats are also known as lipids. And we want to look at WENT4 specifically because it's a signaling protein, and we wanted to see the relationship between WENT4 and how it might be controlling metabolism. So something that Maddie already touched on is that lobular cancers, breast cancers, prefer to eat fats over sugars. They don't really love to eat sugars. And we can see this here. So when we have WENT4 in an overexpression, or when WENT4 is turned on, we have in the cells a decrease in fats that are stored within the cells. And when WENT4 is off, the amount of fats stored within the cells goes up. This is indicating that WENT4 is contributing to those fats stored within the cells to be eaten and consumed, and hopefully, and ultimately generating energy. Oh, wait, sorry, wrong button. Generating energy over here. And so we wanted to understand what is going on with WENT4 that is causing these changes in fats being broken down as opposed to not being broken down. And so a protein that is responsible for mediating several different breakdown processes is AMPK over here. And we can think of AMPK like a button. AMPK gets turned on when we don't have a lot of energy available in the cell. And one of those signalers to indicate that we have low energy is AMP. So AMP will turn on AMPK, and it will start the process of the breakdown of sugars and the breakdown of fats and mediate which of those pathways is being prioritized. And then we get energy in the form of ATP. And so one of the things to help solidify the relationship between AMPK and how it's reacting in different, when these cells are growing in different conditions that are enriched for sugars or fats. We have this one diet experiment down here where we ran a Western blot. And I think Western blots are really great to look at because it's very qualitative. And we can see the expression of what we're looking at. So the bands here are the intensity of the band. The more intense means we have more of the protein that we're looking for. And here it's supposed to be the active form of AMPK. So when we have the cells growing in environments without sugar, we see that AMPK is activated a lot more. And when we also have no sugar and no fats, the signal is even further intensified. signaling that there is not a lot of readily available energy to the cell, and we need to make more energy. So ultimately, this helps us, and we also have data that contribute that is indicative of Wnt4 controlling AMPK as well. And so that's just a bit of what we're looking at and the web of metabolism.
Hi, everybody. I'm Cameron, and I am, oh, how do I change this? I am a research technician in the Sikora Lab, and one of my main focuses right now is in vivo work, so all things mice. I'm kind of the crazy mouse lady in the lab. So I do things related to the experiments, you know, starting up the experiments, giving the mice, inoculating them with cancer, and then treating them and carrying the treatment out, measuring tumors all throughout the treatment, before treatment, during, after, as well as the not so glamorous day-to-day chores involved with keeping many mice, like making sure they have proper water, the right kind of water. As Matt mentioned, we have estrogen supplemented and estrogen deprived water for some of these mice. So that, making sure they have the right food, enough food, weighing them all day, every day. So Matt already mentioned this experiment a little bit, but I think this was obviously one of our main mouse studies that we have going on right now, the preclinical combination therapy with endocrine therapy and PARP inhibitors, talazoparib. So super quick refresher, we have 4 different treatment groups, our endocrine therapy and our no endocrine therapy. And then within those, we have either plus or minus talazoparib. And the endocrine therapy is not an added treatment that we give the mice. It's actually a mimic of an aromatase inhibitor. We just mimic what an aromatase inhibitor would do, which is decrease estrogen. So we just take away the estrogen in the water. So this is one of our examples. We're doing this in a bunch of different cell lines and those PDX, the patient-derived models as well. But this is just one of them. And as a reminder, that purple line is our combination treatment. And it's not only very effectively just knocking down that tumor during treatment, the 6-week treatment, but it's also showing effects after the treatment period, and it's taking way longer for those tumors to regrow as opposed to those single agents or control groups. So yeah, this is very exciting. We're still working on this in different cell lines, as well as that mind technique where we inject the cells directly into the ducts. And yeah, hopefully looking towards clinical trials. This is a little side project of mine. It does not have to do with, it has to do with mice, but not as involved with mice. It's a staining technique that I have been using to try to visualize and analyze mouse tissues. We're visualizing, detecting a gene of interest, and in our case, it is Wnt4, because Wnt4 is a little bit difficult to visualize with traditional staining techniques. So this is called in situ hybridization. And yeah, like I said, it's used to detect and localize a gene. So not a protein, but an RNA molecule. So it's, I don't know if you can see it. I pointed to a couple of them. The stain itself is the pink puncta, pink dots. So wherever you see a pink dot in these, that is an individual RNA molecule of Wnt4. So not a protein, but a recipe for a protein of Wnt4. I'm using these right now, these staining techniques right now, only in mice, because that's what I have available to me. But these are 2 pretty pictures I took of an ovary and a uterus. And I'm still in the beginning stages of this process, but through doing more of these, I can start to ask questions like, you know, localizing the Wnt4 and why Wnt4 is more prevalent in certain organs, number 1, than others, and also in certain parts of, like, within those organs. So yeah, and eventually I would like to do this in human tissue and tumors. So yeah, I'll pass it off to Joe to talk about metastasis.
All right, as Matt alluded to earlier, I'm going to talk about some modeling of ILC. Some of these are new models, but again, I am going to show some pictures of some black and white mice. All the mice that you see are sleeping. We can give them a little gas. They go to sleep for a little bit. So they hold still while we take their picture. And then we can get some measurements from that so we can track their disease. So again, I am going to show some pictures of mice and some tissues throughout the discussion. So metastatic ILC, Matt showed this earlier. This is a great image by the LBCA and shows the major organ systems that are impacted by metastases, but we're interested in some focused tissues, particularly those that are unique to ILC. And for the tissues that are not unique to ILC, what is unique about ILC metastases in those tissues? So for example, Matt talked about the brain and the leptomeninges. IDC, ductal carcinoma, goes to the gray matter, particularly more or less. ILC goes to the fluid and the spaces around the brain, the leptomeningeal tissues. That's something unique that we want to do. And Matt showed the picture earlier that shows that our models recapitulate that a little bit. So we want to know why. Why is it our models go there? And how can we exploit those models to learn things about those sites of metastases or the processes, and then figure out how we can treat those. Ovary and uterus are unique to ILC. We've seen that our models go to those tissues. So this is an important mediator to figure out why do they go to those tissues and how. And bone is one of the other ones, which is my personal interest because that's what I trained in previously. The interesting thing about bone metastases here is that ILC metastases actually make bone, where most breast cancers degrade bone. So it's unique that ILC ones make a little bit of bone. I'm not going to say they're like making tons and tons of bone, but they make more compared to other breast cancers. So how do we study this? We're actually going to go back to slides. Is this the, yeah. So, no. Did it? I'll do that. Thank you. So this is a firefly. So we didn't come up with this technology. Others have come up with this technology, but we can take a firefly, and what makes them light up is this protein in them called luciferase. We can take that protein out and force our tumor cells to express it or to make that protein. When we give them a certain reagent called luciferin, it makes them light up just like a firefly does. So if we put that protein into our tumor cells, our ILC cells, they'll grow green. Now again, this is where I'm going to show you one of those mice. So when we put those cells into a mouse and we have them take a little nap and we put them in this big camera here, this is what they look like. Their tumors light up throughout the body. And like Matt said, we're focusing on those lower fat pads and some of the other ones. But one thing I want to call attention to is this mass up here, which is in the head. That's actually a brain metastasis, which has formed on its own in this mouse. So we know that once those form, that's an important mediator, and we know we need to euthanize these mice because they're going to have signs and symptoms very shortly thereafter. But this proves that we can inject these in a localized place and they will metastasize just like other breast cancers. So as Matt alluded to, we do this through the MIND technique. We can inject this directly into the duct, and that helps to form the entire sequence of events that goes into metastasis as unique. These tumors will metastasize if we put them into the fat pad, but they seem to do slightly different things. So we know that by doing it this way, we're better recapitulating what actually occurs. So we can use this information. This is a graph. We can actually measure these mice over time and figure out how their tumors are growing and use that as a mediator to find out, can we put a therapeutic in, for example, and inhibit growth in those? Can we track growth? Can we track where these tumors are going? So that's the basic model that we're going to talk about. As far as metastasis goes, we have development of a tumor to ILC here. At that point, we have dissemination of the tumor represented by different colored spheres, which are each cells, and they will metastasize through the blood or the lymph. And those actually come back together after a trip through the body. So lymph and blood can become interchangeable at a point, and that's possibly how we're getting some of the brain metastasis in here, and probably the bone. The lung metastasis that we've seen, those are probably a little bit more of a toss-up. We haven't really looked at the direct lymph nodes draining from these mice because that's very difficult. They're very small. They actually don't get big. So it's kind of a transient passage through those structures as the tumor cells go somewhere else. After they go through those structures, they can go to bone, ovary, or brain. And as I said, these are the three that we're going to focus on for this time. as you can see down here, we have two of our ILC models, MM134 and 44PE, and we can see that those metastases develop in the brains of those mice and the bones. So you can see this isn't just one spot in either of those tissues where they develop. They actually consume those entire tissues, and then we can take them out and perform histology on them. We can look at what their genetic makeup is. We can see what genes they're turning on and what genes they're turning off, and a lot of other unique characteristics. We have looked for GI tumors. Oh, I'm sorry. The question was, are we looking for GI tumors in these mice? And yes, we are. The issue is that they're hard to find. They're not particularly prevalent in these mice. We were able to take one out. It looked like it was kind of a pancreas that we were able to pull out. But as for some of these more the omental tissues, we haven't seen a lot of big signal from there, and we haven't been able to track those down at this time. To that end, as Matt said, when we did this originally study, we were taking mice at different time points, but even at the later time points, we didn't see a strong signal from those. Yeah. So as I mentioned, this is the histology. So here, this dark purple part is the tumor, and this is normal brain. So this is what normal brain tissue in a mouse looks like, and we can see that that tumor is crowding out some of the brain. And down here in the bone, we have this tumor, which is a little against what I said before, where the cells kind of make bone. There is some segments here where they are making a little bone on the edges, but this tumor has actually moved through the outside of the bone here, and this is more normal bone. So these do recapitulate what women with ILC develop. And then Matt showed this earlier, where we can actually track how these tumors come up. So what are we trying to do? Now that we have a model, we can start to break this down and find out where we want to intervene, or how we want to intervene, where, when, and how. So I wanted to use this up here. We can start to study these cells that are going into the circulation. We can study the cells in the circulation if we choose to. We can get them as they're coming out and maybe just starting to form tumors in these tissues. And then finally, we can take the tissues out of the established metastases that develop and see what's making them. So as I know before, bone is my interest, and we wanted to look at what is bone. And as I said, the ILC ones make bone a little bit more than other tissues. So this is a culture assay, which we can do outside of a mouse. We took the tumor cells and had them secrete proteins that we could then put on pre-bone cells to see if they would make bone or not. The ILC ones here, you can see with these little black dots, are actually making mineralized bone structures in a cell culture plate. So we can then take that and then look at the mice and go back and forth and say, what are they doing? How are they doing it? And try to define the mediators that are important for those methods. The IDCs, this is completely clear. They don't do it. They actually cause bone to break down. And we're working on defining that as well, so we can compare to ILC to see where the similarities and differences are. We're also able to track the genetics of these tumors. So we were able to grow out the metastases from those mice and then take out their RNA to find out what genes were on and off from each of those metastases. So we could say, oh, these genes are on in bone and they're off in brain, for example. Or these mice have these genes on in their bones, but these mice don't have those genes on in their bones. And what we were able to see is that in general, across bone, ovary, and brain, if a gene is on, it's on across all of them. And if a gene is off, it's off across all of them. So what that's telling us is that whatever is going on here is occurring to all of the metastases. So there's some unique genes that are on and off in all of the metastases, which are important. But we can also look at the fringe here and say, oh, in ovary, there's some things that are shut off specifically in ovary. And what does that mean? And that's what we're looking at right now. And we can create these networks to say, oh, this pattern of genes is on, or this pattern of genes is off. How can we go back and say, is there a drug that we can use to turn that on or off? Or is there another pattern that we can do to say, we need to look here to find a better mechanism to explain why these cells are going to these tissues? And lastly, I'm going to come back to talazoparib thing, because we wanted to talk about intervention. So I think it came up earlier of, can we inhibit disease? And that's what we're looking at here. So we took mice, we allowed them to establish metastases throughout the body, not just bone and brain and ovary. So these mice just made their own metastases, and that's when we started treating them with talazoparib. Now, this data is very early. We've only been treating for about 2.5 weeks at this point, and we plan to treat for 10 weeks. But hopefully we can appreciate that the control and the tala alone mice, their tumors are still increasing when we measure that entire mouse. This is not differentiating metastatic disease from primary disease. So in these mice, all of their disease is increasing. However, the mice that are receiving that AI, that withdrawal of estrogen or the combination therapy, had a pretty stark decrease like we saw in the previous models. So we want to follow these mice out, see if the combination therapy is only impacting the primary disease, or is it working against all of the disease, and how? And how can we use that to go forward and find new targets to treat ILC? Thank you.
All right. Thanks, gang, for presenting. In the interest of time, I'm going to get through my last part, and then everybody will be up at the posters at the end for you to ask questions, learn about their research. But as I said, they're the ones that actually do the work. I haven't touched a pipette and moved anything like that in years. They don't even let me go downstairs to the animal facility. So they're the ones that are doing all the hard work. In this last part here, I want to talk about what it actually takes. This is the part that's actually my job as opposed to what the lab is actually doing to do the work, but getting support for our research, writing the grants and proposals, and getting the funding to do the work. So I actually did a couple years ago with LBCA an advocate chat on laboratory research logistics. So that's kind of the full version of what I'll talk about now, but I go through the whole process of what we think about in writing grants, what the process looks like, getting funding, et cetera. So I'll give you kind of the distilled version here today. But effectively, what my job largely boils down to is coming up with ideas, using those ideas to garner support and to convince people to come work in the lab. And then we use the funding to also buy the stuff we need to do the research. So I need to get the people and the stuff and turn that collectively into the science, which you just saw what these folks are doing. So what does that look like? Who's actually in the lab? And I wanted to just give you a brief rundown of the folks that tend to work in research labs. You just met a lot of the different positions now. But what it actually looks like in a general lab is, so I'm the principal investigator or the PI. My name's on the door and the buck stops here as far as responsibility for what goes on in the lab. PIs are typically PhDs and or MDs. The track goes from assistant professor to associate to full professor. For all of these, I put up salary ranges just so you can get a sense of what we're budgeting, and it'll make sense in a moment why I'm showing that too. I'm kind of going down in seniority here. So after the PI is often staff scientists. These are folks that are typically PhDs. They may be non-tenure track faculty like Dr. Sotnick is. Also, then we have postdoctoral fellows. Maggie and also you didn't get a chance to meet Taylor are 2 PhDs in the lab that are just out of their graduate work, and this is more of a training position after they get their PhD. Labs often have graduate students. These students usually have their bachelor's, they may have a master's degree, and they are actually in training for their PhD. A lot of labs will have clinical fellows who are at a similar stage. They usually have their MD and are in the lab for a year to year and a half. Many of the folks you just met are research technicians. These are full-time jobs after completing a bachelor's or a master's degree, and that is their full-time job to do research in the lab. They're not in a formal training position. And then any lab will have any number of trainees, which will include undergraduates, postdocs, and medical students, et cetera. The reason I'm putting salary ranges up here is to point out that as a PI, I am responsible for all of the salary, tuition, and benefits off of my grants. So that's most of what I'm writing grants for, is to pay the people that work in my lab. We do have to get the stuff to do the research though, and this is kind of a sense of what we're looking for when I'm writing grant proposals to actually get funding. A lot of and this is just kind of pictures of some of the consumables and capital equipment that we'll talk about. We have to buy everything. This includes the test tubes, the flasks, the tips for fluid handling, the gloves, the micropipettes that are the actual fluid handling tools. All of our experiments require growth medium to actually keep the cells alive. One of the weirder things we have to buy routinely is actually bovine serum. This is serum from cows. This stuff is weirdly expensive and it relies on the commodities market. And I could go off on that, but that is a whole process we have to go through in maintaining consistency in supply and dealing with agricultural products, but this stuff's expensive. A lot of purified growth factors and the proteins that keep these cells alive in culture. And then there's a lot more things like antibodies and specialized experimental reagents that we have to budget for. A lot of what you saw data for is from next generation sequencing, which is actually taking genetic material from cells and making it a material that we can sequence. That costs time. And then working in a cancer center, we have access to a lot of fancy equipment that we couldn't afford on our own, but that does cost hourly rates. You can ask Dr. Music about her adventures in flow cytometry and what that looks like. For using fancy equipment. But in general, these consumables will cost around $20,000 to $30,000 per person per year, if I add it all up and average it out. Then there's also the capital equipment. We need these incubators to keep the cells alive. We'll go take a look at these in the lab. If we head over for a tour, these cost upwards of $10,000 each. The biosafety cabinets that create a sterile environment to work in cost upwards of $20,000 each. The centrifuges we use to maintain our cells, et cetera, et cetera. For the animal studies that you saw, immunocompromised mouse currently in academic settings cost $120 a piece. And then we have to pay for housing, which currently, because they're immunocompromised, is quite expensive. And just as an example, one of those studies that we looked at today would have, for maintenance and execution, around 50 mice. Plus, like I said, these are much longer studies for lobular cancer. So plus housing, you're well over $8,000 just in animal costs, care costs. And then there's also the additional costs of the consumables, the syringes, all of the imaging we do costs hourly rates, the chemicals we have to use, et cetera, et cetera. And then it costs us money to publish. We have publication fees. The way that my lab designed to publish, we don't pay the high end. We focus on open source journals. We try to get around this. And then actually go present our work also costs money for registration fees and travel, et cetera, et cetera. Actually, almost the entire lab is headed down to Albuquerque for a steroid receptor meeting today. And we're done this afternoon to go talk about all the work you're going to see in the back. So how do we actually get the funding to do this? I have this broken down into two major sources that I'll explain in a second. New labs, whether it's a startup lab or whether labs move institutions, get startup funding. This, depending on career stage, is anywhere from around $750,000 to $2 million or more at some institutions. We rely in a lot of contexts on philanthropy, and obviously I'll come back to this. This is rarer for PhDs. For MDs, this often comes from grateful patients. I'm really fortunate that my advocate partners established the Lobular Breast Cancer Research Fund, and we'll talk more about that. Where most of our funding comes from, though, are federal and foundation grants. Our federal grants for our lab mostly come from the National Institutes of Health. The Department of Defense also has a robust grant program, as Donna mentioned. We write a lot of grants to foundations like the American Cancer Society, Susan G. Komen. We also in Colorado have the Cancer League of Colorado, which is an outstanding organization for funding high-risk, high-reward research, especially on campus. There's also Vyname, which is a lobular-focused foundation, and LBCA has some of their own grant programs as well. And then some labs can get funding from industry programs. This is usually for clinical trials. There's some contract research for deliverables, maybe for new drugs, less common for those of us doing basic research. These two pools, though, I have broken down. This top one is what we would call unrestricted or discretionary funds. These are not obligated for certain projects, and the PI can use these funds per their lab's needs. But the majority of our funds are restricted. We write grant proposals to get these, so we're obligated to do what we said we would do. We have some leeway around the science, but in general, we don't necessarily have a ton of flexibility for what we're actually spending those grant funds on. And obviously, if we have new ideas or want to go in high-risk spaces, these unrestricted funds are particularly important, which is where the philanthropy comes in. So a lot of what we showed you today was new ideas that I didn't necessarily have a dedicated funding stream for. We were able, for a lot of the studies, to cobble together funding from existing grants where the science does overlap. Doing that always does create some risk as far as completing the concrete project goals. So we have to be very careful about when I decide to do that to kind of take new directions, as long as it's connected enough to what we were proposing. We can write pilot grants. There are a lot of great mechanisms to get new ideas, but sometimes you get into kind of a weird recursive loop where you need data to justify the grant and you need the grant to get the money to get the data. So we get stuck sometimes in those spaces. And there are mechanisms that help us get around it, but sometimes it works, sometimes it doesn't. But this is where discretionary funds can best come into play to chase these new ideas. We were really fortunate that for some of the work I showed you today, that the Women's Cancer Developmental Therapeutics Program has their own pool of discretionary funds from philanthropic support, that they were able to help us to do studies that I would have had to cobble together budgets over the next 2 years. We did like yesterday, which was really exciting with that support. But for us, then the main source we have right now that, again, I'm really fortunate and ever grateful that exists is a Lobular Breast Cancer Research Fund, which is a fund that exists at CU Anschutz that is then under my control to use to support lobular research in the lab. This helps us do large-scale projects that are sometimes outside the funding mechanisms of normal grants, in particular like building patient tumor data sets, which are like logistic heavy and don't fit nicely into proposing new science. But what I want to share today that Carmen talked about was actually what was the confluence of a very timely donation, a new technology that got launched, and an email from Carmen, a student with relevant experience that helped us go after a new research in ILC. Many of you have seen this, but if you'll indulge me, this is the video that we put out for what I've been calling the Lobular Breast Cancer Research Fund in action, and one example of what we were able to do with philanthropic support. Hopefully this. One of the more common and powerful ways that scientists can understand the inner workings of a cancer cell or tumor is to measure patterns of gene expression. That is, to assess how individual genes in the genome are turned on or off. Any cell in the body responds to stimuli, stress, or other cues by changing gene expression to then build the cellular machinery it needs to respond to those cues. For example, changes in gene expression may ultimately cause a cell to grow and divide versus to move or migrate to a different place within the tissue, as either of these events would require very different machinery for the cell. Cancer cells are no different, though the underlying processes have essentially been hijacked relative to normal healthy cells. Measuring gene expression in cells can give us insight into what makes cancer cells different from normal cells, how they utilize cellular machinery to constantly grow, or how they respond and adapt to critical stressors like anti-cancer therapies. Normally, when we measure gene expression, we harvest a large number of cells, either a fragment of a tumor or a large pool of cells grown in the lab, either containing millions of cells. Pop them all open, collect the genetic material, and measure the genes. This approach, usually called RNA sequencing, is powerful, yet the resulting data essentially represents an average across the millions of cells collected. We've learned that cellular populations within a tumor are diverse and may represent numerous functional states, each of which may contain different resistances and, importantly, vulnerabilities. Understanding tumor cell subpopulations has rapidly advanced, down to the level of individual cells. Technologies that are currently available to perform so-called single-cell RNA sequencing use highly advanced microfluidic devices, that is, plastic chips with tiny channels to move fluid, to separate individual cells, which are typically about the diameter of the width of a human hair each. Once collected, these devices can isolate the genetic material per cell for subsequent analysis. Though extremely powerful, these platforms are expensive and require significant technical expertise. With the support of donors to the Lobular Breast Cancer Research Fund, the Sikora Lab was able to acquire a new platform called PIPSeq. Rather than using the expensive microfluidic chips, this system creates an emulsion of cancer cells with chemicals needed for RNA sequencing, not at all unlike emulsifying fats in the kitchen to make mayonnaise. By carefully vortexing cells with this emulsion liquid, a fancy term for controlled, vigorous mixing, we create tiny fluid droplets that each contain a single cancer cell. Once we have these droplets, we can use precise heatings and coolings to perform the chemical reactions needed for sequencing to measure gene expression from each individual cell. PIPSeq is substantially more cost-effective and flexible than other platforms, which will allow us to greatly expand our work in understanding lobular cancer cell biology. Using single-cell RNA-seq, we've already been able to identify that as lobular cells grow and divide, they progress through a state where they are highly vulnerable to a class of FDA-approved breast cancer drugs called PARP inhibitors. Importantly, lobular cancers are typically ineligible for PARP inhibitor treatment, as they overall do not carry any features of PARP inhibitor sensitivity. By looking at single cells, we see that the PARP inhibitor sensitive state is likely only a small population of cells at any given time, but cells likely must pass through this state in their lifetime, and over time, more and more cells will die off in the presence of these drugs. This has led us to test these drugs in lab models of lobular tumors, where we are seeing very exciting responses to PARP inhibition that would have otherwise been missed without these insights. Moving forward, single-cell gene expression studies using PIPSeq will allow us to dissect how lobular cancer behaves, grows, and evolves. We could not have acquired this new technology without the generosity of LBCRF supporters, which also supported our hiring a student that developed the programming and analysis tools to process these large data sets. We're extremely excited to continue this work to bring new treatments to patients facing ILC, and we are ever grateful for our philanthropic supporters. So in thinking about what I kind of explained up front, that we need the ideas to get the funds, to get the people and the stuff, it was quite concrete as far as people and stuff with this particular set of donations that supported this. I didn't have the budget to hire a student at the time, and particularly to take one as high risk, but Carmen had happened to, like, timing wise, reach out about a potential project. So we had the funds to support her, her joining the lab ultimately. And then the stuff was literally a special heating block, that fancy vortexer, the chemical reagents, and a fancy computer to do the actual analysis. So this is not stuff that I had the budget for. This is not stuff that we had. You know, we had ideas to do it, but we're kind of restricted to the older, expensive pipelines. But with this type of donation, we're able to expand into new space, literally and figuratively, with these new technologies that are now available. So this is the current structure for the Lobular Breast Cancer Research Fund. It's a pipeline through the University of Colorado, so you're not donating to me. I'm not using this to go buy a yacht or something. This is actually a CU fund, so the instructions have been updated recently on the page. The fund was initially established by Lori Petiti and Lee Pate, who made a particularly generous bequest to it in her estate. And then this QR code will go directly to that page. And obviously, I'm happy to talk with anybody about what we're thinking about using donations for, and how this is helping move our science forward. But with that, I want to wrap things up for our kind of virtual and presentation program. Thank you all for being here and for giving. Not that we need motivation, but I can't say enough on how it means to us to see the women who are essentially our research constituents, and knowing that you care about what we're doing in the lab. We say a lot in the lab that science is hard for stupid reasons, and knowing that you care about what we're doing means a lot when science isn't working for a lot of stupid reasons. So right now, our lobular work in the lab is supported, in addition to support from BCCRF to actually put out these programs. Our research is supported by NIH, ACS, the DoD, Dynamy Foundation, Cancer League, the Women's Cancer Developmental Therapeutics Program, and the Lobular Breast Cancer Research Fund, as well as support from the Cancer Center here. And thank you to the lab. Like I said, we're now 8 people full-time, which is really remarkable. We're growing. Taylor just started 2 weeks ago, and we're looking at another postdoc joining in January. So we are legion, and I promise we're doing everything we can to learn more about your disease and bring changes to the way your care is managed. So with that, I'm going to kind of end our main program. We can go chat over posters, and then we'll talk about a lab tour shortly. But oh, Tracy, please. See if we can get a microphone.
Just one final comment, as we are all inundated with Breast Cancer Awareness Month and pinkness and things. I think this really sums up why it's important to know where your funds are going, and if you as a patient or as a caregiver want to make a donation or want your money to go to actual research. That's why we talk about pinkwashing of breast cancer, and why it's really important to know the stuff with pink ribbons on it. Where is that money going, if anywhere? And very little of it actually ever goes to research or patients. And so if you really, really want to make a difference with your donations, this is a perfect way to do it. And I encourage people at home and everywhere else to really pay attention to when they're tempted to buy something in the month of October that has pink or ribbons on it. Really ask, where is your money going, and how can I best support the folks that are working on my disease? So thank you.
Thank you, Tracy.